This video is on photosynthesis, but we're actually going to start on the back of this video sheet looking at a structure that makes photosynthesis possible. So that structure is known as a photosystem. A photosystem is a protein complex, and these photosystems kind of act like solar panels. They collect sunlight and help start to transfer it to chemical energy that we can use to make into sugars. So in eukaryotes, these photosystems are found on the thylakoid membrane. In prokaryotes, like bacteria, these photosystems are found on infoldings of the bacterial cell membrane. And we'll talk about that more later. So let's talk about a photosystem. This entire structure right here is an example of one photosystem. So I thought we'd take the opportunity where we had more space to really look at what's in a photosystem. In a photosystem, we have embedded pigments. There's all sorts of pigments that you can find in a photosystem. You can find pigments of chlorophyll A. You can find pigments of chlorophyll B. You can find pigments like carotene or xanthophyte. And all of these pigments are molecules that capture energy from the sunlight and transfer it to this group of chlorophyll A right here. And each of these pigments preferentially captures different wavelengths of sunlight. So what happens in a photosystem? How does it collect energy? First of all, these pigments capture light energy and transfer it via excited electrons to this chlorophyll A dimer, meaning these two chlorophyll A molecules are stuck together in the center of the molecule. So step one here, I'll write it right here. The pigments capture light energy and transfer it via excited electrons to a chlorophyll A dimer, meaning two chlorophyll A's put together in the reaction center. Okay, so within a photosystem, let me focus this up for a second. Within a photosystem, you have an area called the reaction center. So this whole area in the center of the photosystem is the reaction center. Surrounding it are all of these other pigments that are capturing energy from the sun. So this light energy from the sun hits these pigments molecules out here, and that excites electrons, okay, in these pigment molecules. So these electrons, these excited electrons, start bouncing around and kind of running into each other, running into the pigment molecules. Eventually, these excited electrons are going to excite electrons in this chlorophyll A dimer right here, in this area of the photosystem called a reaction center. Those electrons are going to pop off of the chlorophyll A dimer, and they're going to be caught by a protein called the primary acceptor protein right here. Now, the primary acceptor protein isn't necessarily physically above this chlorophyll A dimer, but I'm just using this to represent energy level. The energy of the excited electrons increases until they are captured by the primary acceptor protein, right? So, like I said, these pigments capture the light energy. They transfer it via excited electrons to the chlorophyll A dimer in the reaction center. And not all light is equal here, okay? These pigments right here, found in organisms that do photosynthesis, absorb blue and red wavelengths of light the best, and they reflect green. So if you see a plant and it looks green, that's not because it likes to use green for photosynthesis, to power photosynthesis. It's because it's rejecting green. Okay. So these blue and red wavelengths are being absorbed, and we use this light energy to power photosynthesis. Okay. So like I said, the pigments are absorbing the light energy. We're using it to excite these electrons, which are captured by this primary acceptor protein. And let's write that step in here just so we don't forget it. Okay. Excited electrons. When I say excited, I mean high energy, which should be like a ding, ding, ding. We talked about high energy electrons in respiration too. So these excited high energy electrons from this chlorophyll A dimer are captured by the primary acceptor protein. Okay. So this is what a photosystem is. We're going to flip the paper over and talk about the first step of photosynthesis, which is known as the light dependent reaction, and it relies on these photosystems. So I thought I'd give you kind of the backstory first. So let's open up our big sheet, and we're going to focus right here on this part of the chloroplast. In the chloroplast, we have our outer membrane, we have our inner membrane, and then right here, these membranes are known as the thylakoid membranes. So the chloroplast actually has 3 membranes. So let's get into the details of the first step of photosynthesis. There are 2 parts known as the light-dependent reactions. So the first step of photosynthesis is known as the light-dependent reaction because it needs light in order to work. And it occurs on these thylakoid membranes. The first part of this, and the second part of photosynthesis is called the light-independent reactions, but that will be in a different video. Okay, so the first part of this, how the light-dependent reaction works, will kind of be a review of what the photosystems do. There are 2 main photosystems that operate in photosynthesis, photosystem 2 and photosystem 1. Photosystem 2 comes first in the process, even though it was found second. So it's called photosystem 2 because it was discovered second, but actually it goes first in the process. And it prefers wavelengths of light at 680 nanometers. Photosystem 1 goes second in the process, and it prefers wavelengths of light at 700 nanometers, although they can absorb other wavelengths of light as well. So what happens first? Okay, first, step 1 here, the light energy excites electrons in the pigment molecules. Of photosystem 2, right? So these electrons are bouncing around in there. Okay. Second, the excited electrons from these pigment molecules transfer their energy to this chlorophyll A dimer, like we know, and electrons pop off of the chlorophyll A dimer and are caught by the primary acceptor protein. Which we know, right, is in this area of the photosystem known as the reaction center. So step 2 right here, excited electrons from the chlorophyll A dimer are caught by the primary acceptor protein. So all of that's review. So we have these excited electrons right here. This whole photosystem, you guys, is actually like embedded in this thylakoid membrane. And this thylakoid membrane is made up of a phospholipid bilayer. So it's like phospholipids. Imagine phospholipids all around this with embedded photosystems in there. Okay. Here's part of the problem, though. If we continuously take electrons from the chlorophyll A dimer and give them to the primary acceptor in the reaction center, that chlorophyll A dimer is going to run out of electrons. So we kind of have like something that's happening all the time. So I'm not going to really give it a number, but something that's happening all the time, which is we have a water molecules that are inside this thylakoid membrane. Okay. The inside of the thylakoid membrane, by the way, you guys, is called the lumen. This is the space inside the thylakoid membrane. So what happens is, this step I'm just going to give an asterisk. What happens is something called photolysis. In photolysis, once again, light is hitting the water, okay? Just like light was hitting these electrons in the pigment molecules, light hits this water, causing this water to lyse or split. So this water splits up into protons, which help create a proton gradient in the lumen here, split into electrons, which replace the missing electrons from this chlorophyll A, and the oxygen that's left over leaves as a waste product of oxygen gas. Okay, so photolysis is replacing the electrons that went missing when we bump up these electrons from the pigments to this primary acceptor protein. So if we are going to define photolysis, I would say photolysis is defined as water splitting by light. The electrons replace the missing electrons in the pigments. The protons stay in the lumen, which, as we know, is this area in the center of the thylakoid, kind of analogous to parts of the mitochondria as well. Okay, and oxygen gas is produced. And in photosynthesis, that's a waste product. Obviously, we need it for respiration, but in photosynthesis, it's a waste product. Okay, so we've replaced these missing electrons. Where did these high energy electrons go? Well, these high energy electrons are going to be passed down something that will sound familiar to you, which is our electron transport chain. So these high energy electrons that were caught by the primary acceptor are going to be transferred down the electron transport chain. As there's an exergonic flow of these electrons, energy is released. That energy is going to be used to pump more protons into the lumen, okay? And that will happen all over the thylakoids. And again, we'll get a proton gradient here, which is our source of potential energy for later, okay? So the electrons from this primary acceptor that are high energy, they became high energy with the energy from the sunlight, are passed down an electron transport chain, creating a proton gradient. Those electrons are then transferred to photosystem 1. Now, once that proton gradient builds up, okay, we are going to create a potential energy source that can be used to make ATP. So the next thing that happens is also on this thylakoid membrane. You can see up here and down here, we have an ATP synthase protein. That ATP synthase protein is going to allow these protons to flow through it from an area of high concentration to an area of low concentration. Once again, that ATP synthase is going to spin, and it's going to cause ADP and phosphate to join together to make ATP. So we have this fourth step over here, which again should be familiar from cellular respiration, called chemiosmosis. So one of our intermediate products in photosynthesis is ATP. So let's write down that third and fourth step here. The third step is the electron transport chain. In the electron transport chain, electrons move down an electron transport chain between photosystem 2 and photosystem 1, which powers an H+ gradient in the lumen of the mitochondria, right? Powers the pumping of protons into the lumen of the mitochondria. So we have a potential energy source here. Okay, then we use that proton gradient to do chemiosmosis. In chemiosmosis, okay, the proton flow through the ATP synthase from the lumen inside the thylakoids into the fluid-filled part of the chloroplast, the stroma, okay, powers ATP production. And this is actually known as photophosphorylation, right? Light-powered ATP production. So this ATP is going to be used in the Calvin cycle to power reactions. But we'll get to the Calvin cycle later. Okay, now we've left those electrons kind of languishing in photosystem 1. So let's go back to our electrons. So these electrons pass down an electron transport chain between photosystem 2 and photosystem 1, and they eventually ended up in photosystem 1. Once again here, okay, light is going to hit the pigments in photosystem 1, right? Those pigments are going to have excited electrons, which are bouncing around. Those electrons are going to excite the electrons in the chlorophyll A dimer in the reaction center. Those excited electrons are going to pop up and be caught again by the primary acceptor protein. Okay. And then any missing electrons are going to be replaced by the electrons from this electron transport chain, right? So once again, we have high energy electrons up here because light has charged the electrons in photosystem 1. So let's call that, I guess, step 5 in here. So step 5 in here is photosystem 1 absorbs light and excited electrons are captured by the primary acceptor protein in photosystem 1. Okay. Now, finally, what happens is these electrons are not really passed down an electron transport chain. They're passed first to a molecule known as ferredoxin, these excited high-energy electrons, and then they're passed to an enzyme called NADP reductase. So this is actually an enzyme over here. So when these electrons are passed to NADP reductase, okay, that enzyme comes into contact with another molecule, which we know as NADP plus. NADP plus is the high energy electron carrier in photosynthesis. I always think of like NAD is in respiration, NADP, P for photosynthesis, is in photosynthesis. The P doesn't stand for photosynthesis, but it helps me remember it. So what happens is these high energy electrons are passed to NADP reductase, an enzyme. NADP reductase combines NADP plus with these electrons to create NADPH, right? NADPH will be used in the Calvin cycle to reduce carbon dioxide to sugar, right? So it's a good electron source there. So that's our last step in the light reactions. Oops, step 6. Trying to add an extra step in there. So 6 here is when NADP+, which is the final electron acceptor in the light dependent reactions, is reduced to NADPH. All right. So this is the end of the light dependent reactions. And there'll be a separate video on the light independent reactions, also called the Calvin cycle. But if we think about photosynthesis, ATP and NADPH are not final products. NADPH and ATP are known as intermediate products. They're made to help the next step in photosynthesis occurred, but they're not our goal. They're not our final products. So the main point of the light dependent reaction is we're harnessing light to excite electrons. Those excited electrons are going to do the work for us to allow us to create ATP and NADPH, which are intermediates that are going to help with this next step of